back in 2019, a year ago, you received a CIRM grant, a California Institute for Regenerative Medicine grant, for something called perinatal hypoxic ischemia brain injury. And I wonder if you can bring this narrative forward of the things that you've learned and towards that particular medical condition. Describe it a bit, and where are you headed with that research?
With regard to our recent CIRM grant to try to bring stem cell therapies to babies that have suffered what we call perinatal asphyxia or hypoxic ischemic encephalopathy, a major cause of cerebral palsy, I am probably getting back to my roots. And this is a condition that, that I've been focused on for years, since the earliest days of my having found that the brain has these kind of very plastic cells. The condition is one in which a baby around the time of birth, either hours, days, weeks before birth, or during the birth process or shortly after birth, is deprived of blood flow to the brain and also oxygen to the brain. And there are many different causes for it, but the end result is that parts of the brain are damaged. And some parts of the brain that are ischemic in this way die immediately. Some are merely stunned and are kind of dancing on a knife edge as to, are they going to die? Are they going to get rescued? Now, as I mentioned before, the brain of a baby is very resilient. So some of those cells dancing on the knife edge actually will survive. But in many babies, that is not the case. They go on to die, and there is no cure for that. The best we've been able to come up with over the past 10 or 15 years is something called therapeutic hypothermia, cooling, which really is a marginally effective neuroprotective mechanism, which is a little Byzantine in that what we hope is that if we slow down the entire metabolism of the brain, maybe the damage will be less. But I've always been intrigued with whether we could do something for these babies. Now, in the interim, over the years of trying to understand the language of the stem cell, we found a very powerful attribute of the stem cell. I think, as I had mentioned initially, we all thought, well, the stem cell will replace cells and replace genes and replace connections and things of that sort. Well, in the course of pursuing that, we actually found another very powerful capacity of the stem cell. And that was to rescue injured cells around the stem cell. And I would love to say that we sought out to discover this property. And of course, that's not how science works. What we set out to do, and this was brought to my attention by some very astute postdocs in my lab, what we set out to do was to replace things, to replace dead neurons in Parkinson's disease and in ALS and in spinal cord injury and in all kinds of things. And I still remember the postdocs coming to me after a recent transplant into a mouse model of Parkinson's disease that they had both good news and bad news. Where do you want to hear first? And I said, well, obviously I want to hear the good news first. And they said, well, the animals are doing better. You know, it looks like we've made an impact with transplantation. And I said, well, then what possibly could be the bad news? And they went, they ain't our cells. In other words, the cells that we were transplanting were there. They were abundant. But in this model, they were not becoming the dopaminergic neurons. But what they were doing was protecting all of the dopaminergic neurons in the area that normally would have died, and all the connections were preserved. And we went, wow. And then we started moving from model to model, models of Lou Gehrig's disease, models of spinal cord injury, models of stroke, models of traumatic brain injury, even models of some of these kids' diseases. And we realized what the cells were doing. Another one of their properties, to go back to the roots of what I was always thought, these are part of a developmental program, and part of the developmental program is to maintain balance and homeostasis throughout life. And one of them is to provide neuroprotective support, trophic support, detoxify the environment, create an anti-inflammatory environment, make blood cells grow where they've been injured. We call this effect the chaperone effect or the paracrine effect. And we published this ultimately as the cover story in Nature Biotech in 2002. But since that time, this has been discovered to be a phenomenon of therapeutic action of all stem cells in all organ systems across the board. This protective or nurturing effect of stem cells. And to this day, almost every stem cell clinical trial that exists right now, except for some, is based on this concept. Knowing that I had this concept and knowing how hard it really is, because, you know, as this field was moving along, the field of developmental biology was also moving along, including developmental neurobiology, and we started realizing just how hard it is to make a brain, how exquisitely intricate it was to make all the right connections and not make any of the wrong connections. And, you know, I think I and my colleagues just realized how naive were we that we thought that we could just take stem cells, put it into a void, not know anything about the disease, not know anything about the cells, not know anything about development, and just voila, let things kind of just set it and forget it and come back and see things better. Of course,
we've learned a lot in the last decade.
We learned a lot. And in fact, to the extent that it started to become daunting, that how was I going to be able to instruct my stem cells? Even if they got there 50% of the way, they had to go the other 50%. And how was I ever going to be able to instruct them to do what nature initially did so elegantly, including in kids? But when we realized that there was this other powerful action of the cells, that they could preserve what nature did so elegantly to begin with, maybe we were back in business. We could accept that there's a lot we don't know. We could accept that nature does it better than we can ever do it and will ever know how to do it. But if we go in at an opportune time with the opportune cells in the right region, in the right conditions, we can preserve or remove from the jaws of death what nature already did well. And that's when I realized I can do something for these babies. Right immediately, we know when these babies are born, we know who's in trouble. And these are young, developing brains. There's certain parts of the brain where the cells are dead, and you're not going to do anything about those. Stem cells are not Lazarus cells. But there was this whole surrounding area of cells that could go either way. And if at a well-timed period, we put in our protecting cells, our rescuing cells, into that area, maybe they could prevent these cells from dying and preserve not only the cell types, but the connections. We did a lot of preclinical data that proved to be the case. And then I made the case to CIRM, and will shortly make the case to the FDA, that in a condition where there is nothing, and we know that we can preserve connections, the cells are right, the developing system is right, we can identify the babies that will respond to this mechanism of action, all the logic, all the rationale, as opposed to many other kinds of clinical trials, are there. Let's give it a shot. And
so, Evan, that's incredible. Let's move to the here and now. And the world is facing a once-a-century pandemic, COVID-19. And recently, we were excited to see that you received, your research team, yet another grant from the California Institute for Regenerative Medicine that might have an impact on the coronavirus. Please describe that work. And when I read about it, I was enthralled. And when I talked to you about it, about your use of something called mini lungs, an organoid. Please describe this, if you can, in a few minutes, what this grant means and what you see as the trajectory of stem cell research and COVID.
So one of the areas of stem cell biology that has burgeoned is not only the field of taking stem cells and putting it into areas of disease, but also using the stem cells to model diseases in a dish. And in that scenario, it's not the cells that go into the patient, but the drugs discovered from the cells that go into the patient. And we have now been using stem cells to model diseases. Now, as a neuroscientist, we've mostly been modeling neurologic diseases. But I'm also a neonatologist, and I'm a true advocate of making sure that pediatricians are also trained as scientists. And I had a fellow neonatologist in my lab, a woman named Sandra Leibel, who was very interested in lung diseases in babies. And because we're a stem cell lab, we know how to model diseases, she was absolutely welcomed into the lab, and we started a lung team that Sandra was leading. Mostly, we were actually looking at genetic diseases that babies have in their lungs that can prevent them from breathing. And Sandra was, in the course of studying that for gene therapy, was making mini lungs in a dish. And then we started wondering, well, we now have the literature's most complete and most authentic mini lungs that are out there. And initially we're thinking, what other things can we study? And we're thinking about lung cancer, a lot that's not known there. And then COVID-19 hit. And we realized there was more that was not known about this new virus than was known. We didn't know the cell types it was hitting. We didn't know all the receptors. We didn't know what drugs would block it. We didn't know the downstream effects. We didn't know the degree of inflammation or blood vessel disease. And Sandra and I looked at each other, and independently, light bulbs went off in our head, and we said, we can answer this question. And we set around immediately with our mini lungs and started infecting the mini lungs with both the complete virus and then versions of the virus where we could study the cell types it was hitting, the receptors it was using, what the downstream effects were. We could put in inflammatory cells. We could put in blood vessel cells. And most importantly, thanks to other investigators in our institute who had already done a whole huge screen of FDA-approved drugs, except not on clinically relevant cells, we realized we could also fast-track promising cells to patients by taking the drugs, putting them in a clinically relevant system, and maybe even without animal modeling, go straight into patients. So that's what we're in the midst of doing now. We're on a very aggressive timeline of studying mini lungs and testing drugs. Now, there's one important thing that we can do that almost no other field can do. And by we, I mean the stem cell field. It's known that there's an enormous amount of disease disparity out there, that for whatever reason, men get sicker than women, elderly get sicker than kids, that certain racial groups seem to have the disease worse than other racial and ethnic groups. Now, some of that may be socioeconomic environment, or comorbidities, or toxins in the environment and perturbagens. Well, we can mimic that in the dish. We can make patient-specific mini lungs because we're starting with stem cells. We take cells from the patient, we turn them into these stem cells, and from these stem cells, we make lungs. So now we can have male mini lungs and female mini lungs and African-American mini lungs and Latino mini lungs. And we may or may not be able to find out the source of the disparity. Maybe it is the other kinds of comorbidities that are going on. Maybe it's a difference in the microbiome. Maybe it's that there's certain inflammatory genes and receptors, HLA types, that are more prevalent in one race or another. We can study that, but most importantly, most immediately, when we find a drug that works, what we can do is make sure it should work for all patients. We will find a drug and we will make sure that it blocks the infection in the African-American mini lungs and the Latino mini lungs. If we're lucky, maybe it just means all we need to do is tweak the dosing, or tweak the dosing regime, or add a second synergistic drug to compensate for that. But our goal is not only to treat patients, but to treat all patients. And I think the stem cell field, thanks to CIRM, has made it possible to address a concern that's been voiced there, that when we do get a therapy, maybe it won't work for all patients. Our goal is to make sure if we find something that's useful, everybody, regardless of your background, regardless of where you come from and what you can afford, you will benefit.